All right, good afternoon, everybody. It's Will Moyer here, and I'm introducing the next in our webinar series. This is Screening and Enrollment, how to ensure you have subjects. Just a couple of housekeeping items. If you have questions you want to ask, you can either request to interact live, and you'll be granted the use of the microphone. Obviously, you'll need to use the microphone that's associated with your computer, or if you've called in from a telephone or device. Or you can use the Q&A box that is over on the right-hand side to type in questions, and we will make sure that our presenters address those questions. If you do request to interact live and get your voice onto the voice and face, if you turn on your webcam onto the webinar, it takes about 10 seconds before I can grant you access, but I will try to pay attention and get that up for you quickly. If you're interested in continuing medical education credits, I will put up the link at the end, but it is bit.ly/R25eval, and that will take you directly to the form. So without further ado, I would like to introduce our speakers. First, we will have Dixie Ecklund, who has been the Associate Director of the Clinical Trials Statistical and Data Management Center at the University of Iowa since 2006. Prior to joining the CTMC, Ms. Ecklund was a nurse manager of the General Clinical Research Center at the University of Iowa for nearly 20 years and oversaw the implementation of hundreds of clinical research protocols. Currently, the CTS-DMC serves as a data coordinating center for NeuroNext, the NINDS-funded network for excellence in neuroscience clinical trials, which has implemented 4 protocols, as well as the Clinical Isolate Transplantation Consortium, an NIDDK and NIAID-funded consortium, which has implemented 8 protocols studying isolate cell transplantation. As director of, and next is Marianne Kearney, who is the Director of Research Operations at the Neurological Clinical Research Institute at Massachusetts General Hospital. Ms. Kearney Chase is responsible for overall study operations of the NeuroNext Clinical Coordination Center and ensures that all aspects of the planned clinical trials are conducted in full compliance with Department of Health and Human Services, NIH, NINDS, and FDA policies and regulations. She has been involved in a variety of roles in the conduct of multi-center clinical trials for over 20 years with a focus in project management, regulatory affairs, site monitoring and compliance, and site management, and has worked in both academia and industry. So I will hand it over to Marianne.
Thanks, Will. So thanks everybody for joining the webinar today. Dixie and I, as Will said, are going to be presenting on screening and enrollment and how to ensure you have subjects for your study, which is clearly one of the most important things in any trial. So we're starting off with a little bit of a joke here, and these are slides that, these are cartoons that Dixie always finds for us, which I think is fantastic. So this slide really depicts, someone who's trying to lay out the red carpet for research subjects. And while we want to make patients welcome to join our clinical trials, we also don't want to go too far. There are a lot of regulations about what enrollment is and when to approach patients and when you can collect data on patients. So we're going to go through all of that with you today. So next slide. So first we're going to talk a little bit about barriers for enrollment. So why is it that patients don't want to participate in clinical trials? And there are several reasons. The idea of potentially getting a placebo rather than an active drug often can deter patients. So the fact that we want to do a placebo-controlled randomized study can turn some people off. So that is one reason that people don't want to enroll in clinical trials. Sometimes there are treatments that we're repurposing drugs that are available on the market for other indications, and we're using those in studies. So if that patient can receive that treatment off-label from a physician outside of a study, sometimes they would choose to do that rather than participate in a clinical trial. Sometimes when there's very limited experience in humans with a treatment, if it's a phase 1 study, patients get very educated on this depending on what the patient population is. They often look into these things, and if there's a lot of risks involved and there's just not a lot of data, some people wouldn't want to be enrolled in a phase 1 study. again, when there's the risks of the study that sort of outweigh for that particular patient what any perceived benefit might be, that's again why patients hesitate to enroll in clinical research. Again, this goes back to being the fear of being a guinea pig. Having things tested on human beings, it scares people. So I think that's part of what everybody tries to overcome in research in general, is to not have that sort of connotation to research. Sometimes, depending again on what the disease is, when someone first finds out that they have a disease, approaching them about a clinical trial might be difficult because they might feel very overwhelmed. And in an acute setting, that often does happen. So thinking about that from the patient's perspective when you're approaching them, or thinking about approaching them for a clinical research study. Everybody knows that the informed consent form is supposed to be at grade level such that anyone in the general public should be able to understand it. So if you have too much technical information in your informed consent form, or if you approach a patient or their family with too much technical information, that could again turn them away from thinking about participating in your study. So you want to think about that. There's certainly a lack of trust just in the general public about clinical research, which goes back, I'm sure all of you have gone through CITI training and have learned about the history of all of the horrific things that have happened with experimentation on human beings without their knowledge, such as the Tuskegee study that was done. So there is a lack of trust with research, and a lot of people just in the general public, which hopefully we're trying to change that message these days as well. Sometimes people don't want to participate in a research study because there's too much of a time commitment, too many visits, it's too inconvenient. Depending on what the person's disability is, they may or may not be able to come to a visit, or if they can come to a visit, they may need a caregiver to also come to their visit with them. And that means taking time out of work for themselves, for their caregiver. So if there's too much of that, sometimes that's a deterrent. And there's also out-of-pocket costs. So thinking about how are the patients going to get to the study visits? What are the parking costs at your institution where they have to park? Do they need to stay overnight for multiple visits? How expensive is it to buy a cup of coffee in your institution or to get lunch? So those are also barriers if someone has to stay for a long study visit that really impact patients. So next slide. So what we try to do, and I think most people in research try to do this now, is really thinking about your study from a patient perspective and making your study patient friendly, so that you will be able to recruit the required number of patients into the study. So we really advise seeking real practical advice from the stakeholders, which are really the patients, patient advocates, and study coordinators. Your study coordinators are essential in enrolling in your clinical trial because they understand the patients. They hear on a day-to-day basis what some of the struggles are. So for instance, we have as an example here one of our NeuroNext studies, which was in progressive multiple sclerosis, so the SPRINT MS study. And this was a visit. These are all the procedures listed here were for their baseline visit. So figuring out how long this would take, when you're designing a clinical trial, you want to get as much data as you can, and that's great. But you also have to think about how long will that visit take? And it's going to take different amounts of times at different institutions. So this study was conducted at 28 different institutions across the U.S. So things like, having an MRI done during their baseline visit, depending on where that MRI was done, some of them were done off-site. So they were not at the same place where the clinic visit was taking place. So these visits tended to be 8 to 10 hours long, and that's difficult for a patient with progressive MS. So thinking about this from the patient perspective and ways you can make this more doable and more patient-friendly. Next slide. Another great example of making your study patient-friendly and seeking advice was the first NeuroNext study that we did, which was a study, a biomarker study in spinal muscular atrophy. We were enrolling in this study patients that were children from the age from birth to 6 months old. So really looking at, how are you going to enroll this patient group? Obviously, you're going to approach, families and mothers who have just given birth to a child. And the healthy control arm of this study was a big concern as to whether we would be able to enroll these subjects. because you can imagine, even if there were minimal exams done, you're still drawing blood from a patient and doing some different procedures on them. And if it's a baby, it is tough. So what we did was we went to the PI of the study, who was Steve Kolb from Ohio State. He and his coordinator, Amy Bartlett, who are phenomenal, they went about this and talked to a support group, the Families of SMA, or I think it's now called Cure SMA. They engaged this advocacy group. This advocacy group actually had some travel funds. They made some funds available for these families such that when they had long study visits, they were able to reimburse those patients and those families to be able to stay overnight closer to the institution the night before a visit if they were traveling from long distance. The advocacy group also really understands the family perspective and can give great advice on the protocol, and they can also help to somewhat market the study. This group actually, did talk about it at many of their study team meetings. At their advocacy meetings, and they also sort of reached out to the group of patients who did have healthy children. So if a family knew of a relative or a neighbor who had a child that was born with SMA, oftentimes those were the healthy controls. They felt so fortunate to have a healthy child that they wanted to do something to help research and move forward to find some biomarkers in this terrible disease. The coordinators provide an enormous amount of support, and they really help with recruitment. So really looking at who's going to be recruiting subjects. The PIs, of course, are important, but coordinators really understand the family, and they can have a big part in reaching out to advocacy groups as well. Next slide. So one of the other things you want to do very early on is you want to sort of define your population. It's difficult to define a population and really find just the right balance. You want to have your entry criteria that defines your population. You don't want it to be too stringent or too loose. And so you're really walking that tightrope of finding just the right line. If your entry criteria are too strict and too stringent, then you're not going to be able to find those patients. You're not going to be able to recruit enough patients into your study. But if your entry criteria are very loose, you may not be able to have a conclusion from your study. You want to make sure you know which patients you're treating so that you can actually look for an effect. So next slide, please. So this is a way we tried to look at this with the good, the bad, and the ugly in terms of entry criteria. So here are some, on this slide are some poorly written inclusion criteria. We tried to write poorly. So to say something like adult subjects as an entry criteria, or status post witness stroke, an NIHSS score of 5, and having someone willing to commit to follow-up assessments. The next slide will show you well-written criteria. And this actually comes from the PRISM study. So this is on the web. You can find this, but this clearly outlines, what is adult. In this study, they clarify that it's anyone greater or equal to 18 years of age. They are allowing an NIH score, an NIHSS score of less than or equal to 5, as opposed to choosing a number. They're also allowing for treatment to be initiated after the patient was last known at their last known well time, as opposed to being witnessed, and also sort of describing the consent process and what they need to be willing to do and provide written informed consent. So if we look at the next slide, and sort of, think about what was written poorly and what was written well. So again, saying adult subjects, what is the age of maturity within each state? For some states, it's 18. For some, it's 21. So clearly saying what is adult mean. So identifying what age group. Again, instead of saying a witness stroke, you might want to think about opening that criteria up to saying the last known well time from that patient. Again, it's up to you in terms of what your criteria are and what you're testing as to whether that would work for your study. The other thing, obviously, that opened up was the NIHSS score. to say a specific score of 5 would really limit your patient population. If you're looking for someone, for patients that are less than or equal to 5, that's going to widen up your patient population that would be eligible for entry into the study. So next slide. Next thing you want to do is find out, is your study feasible? Are you going to be able to conduct the study? So first you want to know, if you're looking at this being, even if it's a single center study, are there other people in your own institution that are interested in answering your clinical question? You want to make sure that it's a relevant question to the other investigators who might be recruiting patients either at your own institution or at other institutions. You also want to know, is it relevant to the patient population being studied? So are patients going to have interest when they're approached about this study? That's an important piece. And are there competing studies? So are there also studies that have very similar or the same entry criteria that are going to be recruiting patients at the same time? That's going to determine whether or not you're going to be able to enroll your study. And are there subjects that will actually meet the eligibility criteria? And how do you know if there are, how many might actually enroll in your study? So this is where you need to get a lot of stakeholders to help you find this information out. So next slide, please. So I will credit Dixie with this slide because she came up with the rule of 3s in this case. And this is loosely based, but really there's going to be a third of investigators that are actively engaged and really involved in recruiting subjects in your study. There will be a proportion of sites that they say they're active, they say they're interested, but for some reason or other, they're never quite enrolling a patient, or they're enrolling very few, or they're struggling. And then there's going to be a third that just sort of go missing in action, that really are not recruiting. So, I think what you have to look at is making sure you're being realistic about, if you have 12 centers in your study, how many of those are truly committed to meeting these enrollment goals and enrolling patients and actively recruiting patients, and which ones are your problem childs, and really where you want to focus your energy. So think about, even if you have 12 sites enrolling, you may not have all 12 sites that are going to enroll the same number of subjects. Next slide. So pre-screening is something that comes up as a topic very frequently. What is pre-screening? What does that mean? How can you pre-screen? When can you begin pre-screening? So these are really important issues in a study. So you want to start thinking about looking at, are there patients out there that might meet your entry criteria, to even look through your records to see how many patients do I see. I'm sure that if you've participated as an investigator on a clinical trial, you've been asked to fill out a feasibility questionnaire to say, this is the entry criteria for a study. Are you able to enroll patients? And if so, how many patients that meet these criteria do you see on a yearly basis, monthly basis? So really looking through records and determining how many patients do you have in your population that meet that. When you're thinking about your particular study and pre-screening, you do need to get some type of IRB approval at most institutions. You should check with your IRB. It's usually just the ability to go through, really do sort of chart review or EPIC review at this point, electronic review, to look for patients that meet certain criteria. And look through the charts. You don't want to bring in patients that clearly don't meet criteria and approach those about your study. So you do want to do some level of pre-screening. What data you collect during that pre-screening will really depend. It depends on the study. It depends how it's set up. I think Dixie's going to talk a little bit later about what you might be able to do with pre-screening data, depending on what you capture. Do you want to capture, whether or not if this person did not meet entry criteria just on pre-screening, why did they not meet entry criteria? That might be something you want to look at later if there's a protocol amendment and possibly go back to those subjects. There's also rules on what data can be kept. If someone has not signed consent for a study, you can't be collecting data on them. So you have to really work with your IRB and work with the institutional policies as to how much data you can collect, and when you need to destroy data if you have been collecting data. So next slide, please. So again, here's a great cartoon from Dixie on pre-screening examples. So you see is sort of the long line of people, like the masses of people. So if you're thinking about, people who have, I'll use our ALS example, people who have ALS, there's a lot of patients that might come into clinic, but you're going to pre-screen out only a few that meet your entry criteria, or you're looking at multiple sclerosis or some other diseases. There's a lot of patients that are possible, but what you want to do is sort of narrow that down to see who would you actually approach about a participation in a clinical study. So next slide. So a couple of examples in one of our, again, NeuroNext studies, BEAT-MG, which was our third study in myasthenia gravis. This is one example where, 1,872 pre-screens happened at our sites. I believe there were a total of 22 sites that were eligible for screening and enrolling patients. Of those 1,872 sort of chart reviews that were done, the sites identified 135 possible subjects that met entry criteria. Of those, 62 patients actually agreed to come in for a screening visit and were interested in possibly participating in the study. And of those, 52 patients were randomized. So again, sometimes it takes that huge wide net in your pre-screening to come up with those few that could potentially meet your entry criteria. And then if you see of the 135, really only 62 were interested in and wanted to participate potentially in the study. Next slide, please. So moving on to enrollment, it's very important to understand the beginning of enrollment. Enrollment is when a patient signs the consent form. Once they sign their consent form, they are considered enrolled in the study. They may or may not move on to be randomized in the study, depending on whether or not they meet all of the criteria to be randomized in your study. But research begins when you have the patient sign a consent form. Also very important to define upfront what your expected enrollment rate is. So this is a picture of one of our studies, I think it was the myasthenia study, to look at what we projected, how many patients we anticipated we would enroll each month, and taking that out and mapping that to the timeline for the grant application as to when we would finish recruitment for the study. It's really important to not only map this out, but also look at it. And what we do in NeuroNext, I think we do this in most of our studies, even outside of NeuroNext, is we look at, how many sites are activated, and then of those activated sites, are they enrolling on time? And then what is the projected timeline? And I think Dixie's going to talk about a little bit later, it's important to map this out so you can look at it very frequently in your study to determine if a change is needed. And if so, what change would you want to make to change your enrollment curve to actually meet what your projections are? Next slide. So one of the really important things is how do you select subjects? So we have a series of slides here about sort of avoiding cherry picking. So avoid your bias in approaching potential subjects. It's important to have diversity in your patient population. It really should mirror the population that has that disease at your institution if you're doing a single center study, or the wider population if you're doing a multi-center study. So you're looking for diversity, you're looking for sort of a balance between male and female subjects, you're looking for a balance, ethnically across your studies as well. So it's important to have that as a focus of your studies to make sure that you're not cherry picking subjects and you're picking representative subjects that actually are within the patient population that you're studying. So I think Dixie is going to take over from here and do the next slides. Dixie, I'll switch to you.
I'm ready to go. Give me a head shake. Can you see me? Can you hear me? Okay. All right. So a little bit more on the theme of cherry picking. I wanted to give you examples of, this is from the literature, of studies where it's not exactly cherry picking, but it certainly gets to the concept, and it's nice to look at this over the past. Starting with an article that was in New England Journal in 1991, And this was a 10-year follow-up study from the Nurses' Health Study, which has been ongoing forever out of Harvard. And it was looking at postmenopausal estrogen therapy and cardiovascular disease, and came to the conclusion that from that database, which was all nurses who had participated, that they saw, with current estrogen use, they saw a reduction in the incidence of coronary heart disease. So that kind of became the lore of the land at that time. Next slide. This led to, in 1995, there was an article published in Time, and estrogen was, the be-all, end-all at that point. The prevailing medical view was that all women should stay on estrogen, and part of this had to do with the fact that women were living longer, and so they were seeing things like heart disease, osteoporosis, and it was felt that continuing estrogen therapy would help improve those outcomes. At that time, they also thought, though, that there needed to be more research. This was about the time that the Women's Health Initiative study was launched, which, next slide, leads to publication in 2002, where they had to pull out all the stops and determine that for women, it was not beneficial to continue on estrogen therapy overall, because it did not serve as a primary prevention for coronary heart disease. Now, the feeling was that the original population, although that was a wide population, those were nurses who, granted aren't always the healthiest, but they tend to know what the recommendations are. And including the PEPI trial, which was also one of the precursors, the women that participated in those studies were healthy women who were probably of a wealthier demographic, more adherent with their health care. So the findings were, that it was, they exercised more, they had better diet, those kinds of things. So that probably tainted the results in a way that originally we felt that estrogen, was a beneficial treatment for postmenopausal women, but now it's been determined through the Women's Health Initiative study that that's probably not the #1 treatment for postmenopausal women. Next slide. So we'll talk a little bit more about screening, pre-screening, enrollment, how you get your subjects in and how you keep them in so you can get your study done. We've talked about different tools that help you monitor this, but the concepts are the same. You need to find the right subjects, you need to approach them and get them enrolled and keep them in the study in order to answer the question at hand. So the length of screening can vary from study to study. As we said before, subjects, once they're signed consent, they're considered enrolled. You will hear this term tossed around differently depending on whether you're talking from an IRB perspective or from a statistician perspective, because enrolled equals signed consent, but often enrolled equals randomization when you report on number of subjects who actually received the treatment. So it is important to understand the difference between that. And I know a lot of investigators get into trouble when they report to their IRB about how many people actually went into the study, and they don't include all the people that signed consent, even if they didn't go on to a randomization. But if you have subjects who've actually enrolled, signed a consent, and didn't go beyond the screening period, it's important to look at what are the reasons for that. Why did they decide not to go on? Or was it predetermined by the study? Did they not qualify based on some test that was part of your eligibility criteria? It's important to look at the reasons why that data is telling you, and also to understand that you get incomplete data. For pre-screening and for screening data, it's always incomplete because people only go to the point of collecting and reporting until something is a stop. So, you keep going, but once there's a stop, and you don't continue on with enrollment for that subject. And so you need to consider that when you're looking at your reasons why you're having screen failures, because it might not be all the reasons why that person fell out because you hadn't explored all those possibilities and or collected all that data, because it's burdensome to collect data that you're not going to be using going forward. So next slide. So as we said, you need to approach them, you need to enroll them, and then you need to retain them. And it's important to start thinking about that from the beginning, because missing data is the death of a lot of studies. So keep thinking of retention even from the point of when you're recruiting them, and keep everyone engaged throughout the length of the study. As a rule of thumb, most studies, when you anticipate your numbers that you need to enroll, and think about number of dropouts so that you get the complete data set that you need, a rule of thumb is that your dropout rate should not exceed 15 to 20%. So if you need 100 subjects, you better enroll and randomize 120, because you're gonna possibly lose up to another 15 or 20 that won't complete all the way through. But it's also important to think that if you design a study, enroll a subject, randomize them to drug, and they need to go off of drug for a reason, either for an adverse event or because they have, they just don't like it, they don't want to take it, any number of reasons that this happens. Off drug does not necessarily mean off study. And in industry studies, you'll often see that because they don't want to, continue to collect that data. But NIH-funded trials are particularly because those subjects, once they're in the intention to treat population. They might not make the primary outcome, but there's safety data to be collected. There's other, secondary data points to be collected, and it's part of the whole profile of that drug or that study. So even if someone does go off of drug, it may be important to consider keeping them on the study to collect further endpoints. Next slide. So you've started your study, you have your pre-screening, you're starting to screen, and things just aren't adding up. You're not keeping on your linear graph line for enrollment. So it's important to know when it's time to change. Next slide. So this is an example from one of the NeuroNext studies. This is Rhapsody. This is our fourth study, which was an acute stroke trial. And it was a neuroprotectant, it's still ongoing. It's a neuroprotectant drug in addition to TPA. And so this is one of the early enrollment graphs that we have for overall randomization. And you can see from there that we started enrollment in October of 2014. And we got to about June 2015, and we'd enrolled less than 10 subjects, and we were way below the line there. We had expected 23 randomizations, and we had only randomized 8. And this is at about 20 centers across the United States. Everybody was up and pretty much activated in October, November, because with the network, we're able to bring all the sites on pretty quickly. The line and calculations are all based on, expected numbers per month per site once they're activated, and we were having trouble. So next slide. Throughout the early 2015, we were having lots of information from the investigators about a change in standard of care. And in June 2015, there was a publication in the New England Journal about the SWIFT Prime trial. And the conclusion was that patients receiving IV TPA for acute ischemic stroke had better outcomes at day 90 if thrombectomy was done with the stent retriever within 6 hours. So there's lots of things that can be discussed about this because some centers use a lot of stent retrievers, others don't, but it was becoming a practice that was pretty common in all of our sites. And most of our sites, or the protocol actually prohibited people from getting the study drug if they were going to have intra-arterial therapy. So next slide. So we gathered a lot of information, and we were kind of ahead of the curve because that publication didn't come out until June 2015. But through June 2015, we started working with our investigators and made a protocol change. And you can see right there in June, July, where we'll have the arrow, that's when the protocol change went in, and that's when the line started going directly up. And by November 2015, we were on track with our enrollment. We had expected 43, and we had 43 randomized. So this was a very good example of a successful way to monitor standard of care, implement protocol changes. All of this, has to be reviewed in conjunction with the science and the safety and all these things. It's not a simple change in a study like this, but it did lead to us being successful in recruitment, and we've stayed right on the line up to now. So hopefully we will by January have finished our recruitment. Next slide. So, in discussing what we needed to do for that particular trial or any trial that you might be working on, what are the first steps to know if you can improve your numbers for screening or for enrollment or for retention? And the first thing to do is consult with your experts. So, who are the people who are the boots on the ground that know exactly about it? If you're within your own institution and working with a couple of other investigators at your institution or nurse coordinators, be sure you're talking to them about the reasons why people don't want to join your study. If it's multi-center, you're working with a couple of centers, of course, you need to engage those enrolling physicians and coordinators to understand what the barriers are. If you have a steering committee, it's good to go back to that group and get input, because those are presumably experts in your field that would understand your patient population and understand the design and operational questions. And of course, also be talking to your patient advocate, because patients who have participated in trials or are interested in trials or are living with the disease, whatever you're studying, can give you a lot of input into why are the reasons they would not join that study or would not stay within that study. You also need to look at the operational or logistical problems that might be getting your study off track for enrollment. So startup activities, which I kind of alluded to that on the earlier slide, that getting sites all up and running at the same time is easier within a network. And now NIH will be requiring that for phase 3 studies, that you have a central IRB and things like that make all those operational issues easier. But it does help to keep your recruitment on track. There might be regulatory issues that delayed your startup. There might be drug supply issues, other things. But those are all the operational details that go into getting started up, which has to happen before you can start recruiting your subjects in. Next slide. So the slides that Marianne showed, like for the MG study where we had pre-screened over 1,800 subjects, we actually had the sites enter that information into the EDC. Entering pre-screening data is kind of a questionable thing to do depending on the importance of it. For a study that's going to recruit early, quickly, and fast, it's more burdensome for coordinators to actually enter in all the charts that they looked at and document the reasons why they did not enroll into or approach that particular subject for participation in the trial. But myasthenia gravis is a particularly difficult disease to recruit subjects into trials. And so it became important that we really were able to tease out those reasons. And part of the same type of thing that happened in the Rhapsody trial happened in the Beat MG trial. We did do a protocol amendment, and it had a lot to do with the accompanying therapies that subjects were on that had been prohibited, and we collected that information and looked at the safety and looked at several other indicators that were coming from the pre-screening data about why we were not finding eligible subjects, and made a protocol amendment there also. But it's laborsome for study coordinators to enter, do chart reviews. It's probably the least favorite thing that any study coordinator will tell you. That's their list of duties. And then to actually enter it into an EDC is another step. There are some reasons to keep some of that data, because to collect for eligibility reasons, And also, you don't want to approach the same subject twice if the reasons, if you are actually approaching them and they turn you down. But pre-screening data is kind of messy. It's hard to know what to make of it unless you really see a good trend. And there are decisions that need to be made on, how much of it you're going to collect, how much of it you're going to analyze, and what it really means. But it can be useful in many studies to give you an idea of where your recruitment goals are going. Next slide. We talked a little bit about screen failure rates. So, there's the pre-screens, the reasons people don't even get approached. Once they get approached and consent, there's the screen failures because they don't make it from the consent process to the randomization process. And then there's the dropouts. So there's really three, apparently I think in threes all the time. There's really three areas where you're going to lose your subjects. The screen failure might have a lot to do with the criteria that you've set up. So they've consented, they're willing, but their lab values are, if the windows are too tight. You're doing a diabetes study and everybody has to have a hemoglobin A1C and they're diabetic and less than, 5.5. Probably, that's a pretty limited pool if they're insulin-dependent diabetics. So you might want to look at your criteria if the screen failure rate seems to be pretty high, and then accommodate any protocol changes that might, again, still lead to a good scientific question. The reason people have tight criteria is to get the best answer to their scientific question, but there are times when you need to look at that if those things need to be loosened up a little, if it's not going to hurt the science or the safety, and help you recruit better, because if you can't get to the end of the study, you don't have an answer either. So next slide. Again, third step is looking at the dropout rates. So you have subjects in, they've screened through, they're randomized, but then they're dropping out. And a lot of times that can have to do with it's just too much for them, particularly if they have a disease. Alzheimer's, ALS, there's often a caretaker who has to accompany the participating subject. So, there's lots of factors that should be considered on if you're losing people throughout the course of the study, and you can look at if there's protocol changes that could be done to help get to that, because missing data, coming from the data coordinating center perspective, is again the death of a study. If you cannot hold up your outcome at the end of the day and you have too much missing data, then there's some statistical tests that can be done to help to accommodate for that, but it's critically important that you get as much data, if not all data, on all subjects through the end of the study. Next slide. Okay, this Marianne and I wanted to include a little bit, and probably most of you are familiar with CITI, CTTI, which is the Clinical Trials Transformation Initiative. It's out of Duke. They have a lot of tools. They have great websites. This particular recommendation came on efficient and effective clinical trial recruitment and planning. I actually served on one of the advisory panels for this back in 2014 or 15, I think. But they released these in May of 2016. That's the website for the recommendation. And it also includes tools with it. And they're listed there. There's a decision tree. There's a stakeholder analysis tool. There's a tool for monitoring your recruitment and performance. And then there's also a note about patient-reported outcomes. Next slide. And they also included this nice framework, which kind of help. It goes over all the concepts that we just discussed through the slides. But as you're setting up your study and thinking about your strategies for recruitment, you need to consider all these three pieces, and including how you design the trial and develop the protocol. Whether your trial is feasible and whether your sites are going to be able to help you recruit effectively, and then what the planning is for communicating recruitment. And we didn't necessarily go into all the marketing things that can be done, but there are certainly things that help sites to recruit and subjects to stay in. And those include things like, birthday cards, newsletters, the super babies that Marianne showed you. All the babies that were enrolled got a little onesie that said, "I'm super." And parents can look at that and think about that, and that might make them a little more willing to go to that next visit. There are always lines that can be crossed, which that very first cartoon talked about. You don't entice people to the point that it might be coercive, but it's nice to be recognized. And I think one of the biggest things that patients feel like, and CITI has actually done a lot on polling patients and advocates and clinical trialists on what motivates people to participate in clinical trials, because it's certainly become a huge industry in this country over the last 30 or 40 years, and it leads to important developments in science. So, but there's lots of trade-offs throughout all this, and recognizing the human subjects protections, but also designing trials that will answer questions and getting them done so that science gets furthered. Next slide. And this is my last cartoon. So, and as I just said, the ethics and the IRB regulations are a part of all those things. But what gets rewarded is when you ease your recruitment requirements, get your trials done more quickly, and the science moves ahead. I think we're up to questions.
That's fantastic. Thank you for those excellent presentations. So if you want to be heard, you can press the raise hand button over on the right to interact live, or if you type your questions into the Q&A box over on the right, we will start to ask them. One question that I had was that in the Rhapsody trial, what was the what was the initial thinking regarding not including patients who were doing the endovascular treatment, and how was it sort of revised as the data came out as the trial went on?
Well, I think the science hadn't really been solid enough that it was a safe procedure to do with TPA, because I think MR Witness came out right before then too, and we were starting to see some of that data. But then with Swift Prime, I think that was a very well described study that was all announced at the Stroke Council that spring, and it seemed that it became more acceptable as a standard of care at that point. I think there's still institutions that's not their first thing to do.
It also, one of the problems with neuroprotection trials in stroke is that TPA and endovascular procedures work fairly well. Therefore, in many cases, there's not additional opportunity to improve for the patients, because many of them are improving without the treatment. So it makes it harder to find those patients who aren't going to improve so you can improve their outcomes. So it ends up being a little bit of a challenge within a challenge. And sometimes when we design trials like that, we try to exclude some of those patients because we think that the treatment is going to really improve their outcomes and there won't be variability enough to see the new treatment actually exerting an effect. So yeah, I think that was quite interesting, but it was very effective, the protocol changed. Unless I am a faulty moderator, it appears that we have left the participants speechless. or they have expressive aphasia. So which is fine. I think you covered things all very nicely. Certainly, we can get them your email if they have additional questions, but truly a delight to have some true experts who've really seen it all in clinical trials. Oh, there was one other thing that I was thinking of as we went through that maybe I could ask one final question. If anybody else thinks of anything while I go through, please type it in. When you're doing an IRB application, one of the aspects of research in screening, and maybe this is almost the pre-screening you described, is actually seeking a waiver from your IRB and your privacy board to search through the medical record for potentially eligible patients. I know that we do this for emergency studies to like screen the electronic health record so that we can start seeing if people are coming in with certain conditions, knowing that it's not feasible to consent people to that part of screening, but you have to receive, you have to seek and receive an exemption from sort of consent to do the pre-screening, and then obviously the patients are consented through the standard procedures after they're identified from that. I guess, do you have experience with those sorts of situations where an element of your screening is done in the background through a HIPAA waiver and an IRB waiver, but then there's the additional engagement once you start approaching the patients?
Yeah, so I can address that. So yes, we do see that on most of our studies, and that's usually in the pre-screening stage. So most institutions will allow a waiver, as you say, to be able to look through the chart and look through for potential patients. And so that's usually when that's done. It's when the patient gets approached that there would need to be consent. So as you said, Will, that's really the pre-screening stage to be able to look through the charts and see what patients are coming in with that type of disease. Dixie, anything to add to that?
Right, and that's just part of your IRB application. You talk about your trial, and then, but one of the requests in your IRB application is that you need to tell them what you're going to pre-screen, what particular data you're going to look at, and then as they approve the whole trial, they will also hopefully approve your partial HIPAA waiver to look through medical records to approach subjects.
Yeah.
And again, it may be your IRB may vary. They may call all of that screening and not delineate that, at least in the nomenclature, of pre-screening versus screening. So it's sort of like two different phases of screening. So as you can see, that might become confusing. The concept is exactly as Marianne has laid out, but your IRB may call all of that as screening. And just make sure, as long as you draw the little timelines and you see what's happening when, it should be clear to everybody. But it's one of those opportunities for confusion. And the less you can confuse an IRB, the better. You know what you're doing, and what you're doing is ethical, and it just has to go through the right channels. But I like the term pre-screen versus screen, but your IRB may not use those terms. So just see what they call it and make sure they understand what you're doing and when.
Good advice.
Well, with that, I greatly appreciate the time you guys have put into this presentation. I think I should be able to find a little way to put up the evaluation link, which is bit.ly/r25eval. I will. I think I can put that into. I don't think I can write myself a question. Usually Sam is able to run this, but if you just remember, if you email me if you miss it, or just remember bit.ly/r25eval. And if you want CME, there's a method through that to get CME. So again, thank you very much for spending a little of your afternoon with us, or if you're on the West Coast, or even in Iowa, it's just turning to noon now. your morning. I hope everybody else has a good rest of your day.
Thanks, everyone.
Thank you.